Bismillahirrahmanirrahim, and welcome back to this next video. And this is another video in the series of videos on the isotypes of antibodies. From the last couple of videos, we were discussing about an important isotype of the antibodies, and that was immunoglobulin G. We had a detailed discussion on the immunoglobulin G and its subtypes, the immunoglobulin G1, the immunoglobulin G2, G3, and G4. Now, in this particular video and in the videos to come, I want to focus on another important isotype of the antibodies, and that is known as the immunoglobulin A. Before going into the immunoglobulin A, let me give you a general introduction to the antibodies. The immunoglobulins and the term antibodies, they are usually used interchangeably, and the immunoglobulins, they are glycoproteins in nature. What I mean by that is that when these immunoglobulins, they are made in the protein form, they go through the process of the glycosylation in the post-translational modification, and in this particular mechanism the carbohydrate moieties, they are attached to these immunoglobulins, which are actually protein in nature. So when you attach the carbohydrate moieties to the proteins, we term them as the glycoproteins. So the immunoglobulins, they are glycoproteins in nature. These immunoglobulins, they are produced by specialized type of the B cells, which are known as the plasma cells. These antibodies or the immunoglobulin, they are part of the adaptive immune response, and what they do is they are responsible for recognizing and neutralizing the foreign antigen or other harmful particles in the living organisms. If you look at the structure of the antibodies, if you generally talk about the structure of the antibody, so you are going to see two heavy chains and you are going to see two light chains. And within these chains, you are going to see the variable regions. These variable regions of the antibodies, they are responsible for binding to the antigen. And the other important region that you see in the immunoglobulins, they are known as the constant regions. And these constant regions, which actually make most part of the immunoglobulins, they are responsible for interacting with the other components of the immune systems, like the macrophages, the neutrophils, which are the immune cells, or they can also interact with the complement system. Now, these antibodies, they are divided into five major isotypes. They are also known as the five major classes of the antibodies, or they are also known as the five major isotypes of the antibodies. In each of these classes, they have got distinct structural and functional properties. Now, how you are classifying these five major isotypes into like these different classes? Now, this classification or these isotypes, they are classified based on differences in the constant region of the heavy chains. So the differences in the constant region of the heavy chain, that is actually the base for classifying the immunoglobulins into these five major isotypes. Now let us talk about the immunoglobulin A. Now the immunoglobulin A is a monomeric antibody, and it contains two heavy chains and two light chains. So you might be thinking that you have read somewhere that the immunoglobulin A can also be found in the dimeric form. If you are thinking so, you are absolutely right. The immunoglobulin A can be found in the monomeric form, but they can also be found in the dimeric form. But in this particular video, I am focusing on the monomeric form of the immunoglobulin A, and in the videos to come, I'll be talking about the dimeric immunoglobulin A as well as the different subtypes of the immunoglobulin A. So don't worry about that. The immunoglobulin A, just like the other antibodies, have this classical Y-shaped structure. So if you look at the structure of the antibodies, it usually looks like a Y, and this is the classical shaped structure of the antibodies. Now, if you talk about the immunoglobulin A, as you can see over here, starting from here, moving up to here, excuse me, this is one light chain, this is another light chain. So what that means is that the immunoglobulin, they've got two light chains. At the same time, the immunoglobulin A have got two heavy chains. Starting from here, moving down like this, and up to here, there is one heavy chain. Starting from here, moving down here, this is another heavy chain. So the immunoglobulin A have got these two heavy chains. So we are talking about the monomeric form. So in the monomeric immunoglobulin A, you are going to see two light chains. You are going to see two heavy chains. Now, as the classification of the immunoglobulins that is based on the heavy chains, so the chain which make the immunoglobulin A, that particular heavy chain is known as the alpha heavy chains, and because of this alpha heavy chain, they are known as the immunoglobulin A. Now, the molecular weight of the alpha heavy chain that is approximately 70 to 75 kilodaltons, and the molecular weight of the light chain that is approximately 25 kilodaltons. So, if you talk about the tetrameric quaternary structure, so that is nearly 200 kilodaltons because this is 25. This one is, say, for example, 75. So 100 is coming from this side. This is one light chain. The molecular weight is 25 kilodalton. This is another heavy chain. This is 75 kilodalton. So this quaternary structure is actually about 200 kilodaltons. When you talk about the light chain over here, so you are going to see a variable region. This V is for the variable. And when you put an L over here, that means that you are referring to the variable region of the light chain. So when you talk about the light chain, there is one variable region, and this C is referring to the constant region. But when you put an L over here, that would mean you're talking about the constant region of the light chain. And in the light chains, you are going to see only a single constant region, and that is known as the CL. So in the light chain, you are going to see one variable and one constant region. Now the things they are different in the heavy chains. If you look at the heavy chains over here, say for example, this is the VH. So again, the V is for the variable. This H is now referring to the heavy chains. So the H and the L, they are actually showing you that which variable region you are referring to. So if there is an H, that would mean you are referring to the variable region of the heavy chain. If you are using the L over here, that would mean you are referring to the variable region of the light chain. So just like the light chain, in the heavy chain, you are going to see one variable region, but you are going to see three constant region when you talk about the monomeric form of the immunoglobulin A, or when you talk about the dimeric form, because then we are going to add them up. So don't think about the dimeric immunoglobulin A at this particular time. Just focus on the immunoglobulin A. So there is the three constant region. This is one constant region, this is another constant region, and this is another constant region. So they are three. And as you can see over here, is the C I told you, it refers to the constant region. This H is referring to the heavy chain. So this is the constant region of the heavy chain, and this is the third constant region. This one is the second constant region, and this one is the first constant region. Similarly, this is true for the other heavy chain as well. Now, the light chains, when you talk about the immunoglobulin A, that can either be the kappa or the lambda chain. So these are the two most commonly light chains that you are going to see in the antibodies, in all classes of the antibodies. So when you talk about the immunoglobulin A, you can expect the presence of the kappa as the light chain. You can also expect the lambda as the light chain. So this light chain could be lambda, or this could be kappa in nature. Now, when you talk about the antibodies, one of the most important thing about them is their structural integrity. How these immunoglobulins, as they are responsible for recognize the foreign pathogens, the foreign dangerous material, so how they are going to achieve the structural integrity then? And the answer to this is they have got disulfide bonds, and these disulfide bonds are actually giving the structural integrity to the immunoglobulins, in all of the immunoglobulins, and in this particular case to the immunoglobulin A. Now, where these disulfide bonds they are present, these disulfide bonds they are present between the heavy chains. You can see over here, these are the disulfide bond. This is one heavy chain, this is another heavy chain, and you can see over here, you can see these disulfide bonds connecting the two heavy chains to each other. Now, this particular region that you can see in the spring form, this is known as the hinge region, and these disulfide bonds they are present in this particular hinge region, providing structural integrity to the immunoglobulin A. These disulfide bonds that are also present between the light and the heavy chains. So this is the light chain. This one is the heavy chain. You can see these disulfide bonds over here. And these disulfide bonds, they are providing the structural integrity. Thirdly, these disulfide bonds, they are also present between the VL and the CL domain. So this is the VL domain, this is the CL domain, and they're also connected to each other by the disulfide bonds. So the collection of these disulfide bonds between the heavy chains, between the light chains, and between the heavy and the light chain, that is responsible for the structural integrity of the immunoglobulin A. Thereby, it can perform its immunological functions. When you talk about the structure of the immunoglobulin or the antibodies, you need to recognize some of the important regions in its structure. Now, one such important region that is known as the fragment variable. Now, I'm discussing these because most of the time the students, they are not getting the idea what these terms that we use for the structure of the antibodies that mean. So one of the important region that is known as the fragment variable, and this fragment variable, that is actually the variable region of the heavy chain and the variable region of the light chain. So this particular region, this VH plus the VL, that is actually known as the FV region of the antibody. Now, we are using another term when we discuss the structure of the antibodies, and that is known as the complementarity determining regions. Now, most of the time, the student, they confuse that the FV and the complementarity determining region, they refer to one and the same thing. They are right to some extent, but there are differences between the two. So the CDR, they are actually the specific hypervariable regions within the FV region that directly bind to the antigen. So what that would mean is that the CDR, that would be a subcomponent of this FV region, and that is very much variable, that is hyper variable, and this hyper variability is actually leading to the diversity of the antibodies. So you need to understand the difference between the FV and the CDR. So the FV would be the VH plus VL, and the CDR, that would be the region within the FV. And there are usually 3 CDRs in the VH, and there are usually 3 CDRs in the VL. Then we use the term FAB. If you can look at this one, this FAB is starting from here, moving up to here. So this fragment antigen binding, which is known as the FAB region, that would be the FV region. So that would be the VH plus VL, plus it is also covering some of the constant regions of these particular antibodies. So the FV, that would be the variable region plus the constant region that is making the structure of the FAB. Not all of the constant region, only some part of the constant region of the light chain and some part of the constant region of the CH1 of the heavy chain. Now, what this term FC means, if you can see, it starts from here, it moves down up to here. This is known as the FC region, or this is known as the crystallizable fragment. And this crystallizable fragment, what it do is that it is going to interact with specific cell surface receptors, which are known as the FC receptors. These FC receptors are present on the immune cells like the macrophages and the neutrophils. And this FC is also responsible for the activation of the complement system. So what you can see over here is that the immunoglobulin, they are actually making two kinds of the interactions. This FV region, that is responsible for interacting with the antigen, and this FC region is responsible for interacting with other immune cells or other immune components of the cell or the body. We will continue our discussion in the next video, and we will talk about the dimeric immunoglobulin A in the videos to come.